I'm here with Bernard Thorens, professor at the University of Lausanne and awardee of this year's Claude Bernard Lectureship. Can you tell us a little bit about your background, your career, and what got you interested in diabetes?
Okay, this is a long story. No, I'm a biochemist by training, and I've been working doing my PhD thesis at the University of Geneva. And I started a little short postdoc still at the University of Geneva, and then I went to the United States to work at the Whitehead Institute for Biomedical Research at MIT. And I was in the laboratory of Harvey Lodish, where they were interested in cloning, characterizing the membrane proteins. So there are proteins that are there to make the exchange between the outside and the inside of the cell. And so I had a project to clone glucose transporters. And so this was really the beginning of my career in diabetes research, when I actually cloned and characterized the glucose transporter that is expressed in the pancreatic beta cells. And of course, this is the protein that controls the uptake or the entry of glucose into the beta cells, which is the first step in the pathway that leads to insulin secretion. So this was the start of my career in the field.
And how did you go on from there? I mean, today you're doing a lot more things.
Right. I stayed for 5 years there in the US, and then I came back. And when I was still interested in cloning some important membrane proteins expressed by the pancreatic beta cells, because really nothing was really known at the time. And when I came back to Switzerland, I cloned the GLP-1 receptor. And of course, this was something very interesting and very important for, as you know, development of agonists for the receptor to treat diabetes. But this was not what I did in my lab. And so I continued cloning also the GIP receptor, the human GIP receptor, and then working based on this availability of new tools, actually, to study how, on one side, how glucose influences beta cell function, and on the other side, how glucoincretin influence beta cell function. So from that, I started 2 different lines of research on the action of glucoincretin hormones in the beta cells and the mechanism of glucose sensing by these cells.
Yeah, very interesting. So this is also what you will talk about in your lecture.
Right, I will describe that, and also what the work that we are doing now. And so we have several lines of research in the laboratory, but related to these 2 aspects of the control of beta cells. So we have continued to try to understand intracellular pathways that are controlled by glucoincretin and that regulates the beta cell mass and beta cell function during diabetes, and what are the pathways that could be maybe other intracellular pathway that can be targeted to improve beta cell function. That is one aspect, and the other aspect is because this glucose transporter that we cloned, we found out that it is expressed and associated with other cells that are able to detect variation in blood glucose concentration. And especially what we are doing now is to look at neurons in the brains that express this glucose transporter and that are sensitive to blood glucose variation. And this neuron now controls several physiological functions, like the counter-regulatory response to hypoglycemia, but they also control feeding behavior. And these are we are more into now neurobiology, actually, to see how the brain control feeding behavior, but also glucose homeostasis.
Mm-hmm. How do you think it would be possible to get more people interested in diabetes, I mean younger people, to go pursue this kind of research and career?
I think there are many people that are already very interested in that. And so actually we have to attract them and make also the things that we are doing interesting. And I think they can get that there is really interest, that we are actually really basic research. So we are really at the frontiers of what is known in the mechanism of the control of glucose homeostasis. And we are opening up, I think many labs in the world are opening up new ways of understanding the control of glucose homeostasis, which involves a lot of new technologies, could be big data, it could be neurobiology, it could be behavioral studies. And we have to combine all these things together. So I think this is a big challenge. So, I mean, if new students are interested in tackling big challenges, so I think this is really an important and very interesting field of investigation that could be, followed.
Yeah, making more careers like yours.
Yeah.
Is there anything you could recommend to them if they want to advance their careers? Any advice?
Well, the advice, the problem with basic research is difficult research. It's tough. So you really have to be motivated. And once you are motivated, you have to do your work and believe in what you are doing, and then work hard until you get, to do that. But also you have to discuss, talk to people, interact, get support from mentors who can help you, discuss your ideas, your career plan and things like that. So this is also important. So, but I think now, if we consider, research in diabetes in Europe, there are a lot of opportunities, and there is a lot of support and funding also by the European Union for working in this field of research, and also at the frontier between diabetes research, but also genetics, omics, big data, and there are new things that are coming, and this is really fantastically interesting, and it's a big challenge for the future. Yeah.